Hi, in this video we are going to perform the analysis of factorial experiment utilizing library AgroR in RStudio. As far as factorial experiments are concerned, they are those types of experiment in which different factors are studied with equal or unequal precision simultaneously in a single experiment. So for that we'll be utilizing two types of data sets which are already imported on my R console. The first one is Factorial CRD and the second one is split plot design. Since in Factorial CRD this is treated as a traditional factorial example. For a traditional factorial experiment, we have two factors, shade, spacing, and we have a response variable that is known as biomass, and we have another experimental data that is for split plot design. We have blocks, or we can say replications here. We have main plot, subplots, and the study variable here is yield. So we will start with the first one, that is Factorial CRD. Since it is already imported on my R console, in order to see the names or the labels which I have given to my data set, I will simply write name and followed by the name of my data set, that is Factorial CRD. And in order to perform the analysis of this design on this particular data set, simply I will write a function, name of a function, anything you can write, suppose fit, followed by the assignment operator, or you can write the is equal to sign, then with. This is an inbuilt function which is usually to evaluate an expression, and followed by these brackets, and within those brackets I have to write the name of my data set, that is Factorial CRD. Okay, then I have to write a default function FATDIC2, which is a default function to perform the factorial experiments utilizing this library. Then I have to write the name of my first factor, followed by the name of my second factor, and followed by the name of the response variable, which is biomass. Then click on the enter, and there we go. We have all the results available here, starting with the assumptions of the basic analysis of variance regarding errors, that is whether our errors are normally distributed, whether there's an existence of homogeneity of error variance, and whether the errors are independently distributed or not. And you can see all these three assumptions as far as normality is concerned, or the homogeneity of error variances are concerned, and independence of errors concerned, and they utilize certain kind of statistics. As far as normality of errors is concerned, it is to be tested by means of Shapiro-Wilk test, and it has to be non-significant also. It should be always non-significant because it assumes a null hypothesis the distribution of errors is normally distributed. Since here, as the calculated p-value is greater than the 5% level of significance, therefore the hypothesis is not rejected. Therefore the errors are considered to be normal due to the non-significant p-value. And in case of Bartlett's test, we have a non-significant test here. Again, the variances, error variance, are considered to be homogeneous. And as for the Durbin-Watson test is concerned, it is also found out to be non-significant. And we have a clear evidence that the errors can be considered independent. That means errors are normally distributed as well as independently and identically distributed. And we have an additional information here, the overall coefficient of variation for our fitted model for this experimental design in a factorial setup. And we have a general mean here, then median, and there is no existence or the presence of outliers. Then we have a traditional analysis of variance table here, for we can see the sources of variation for the first factor, second factor, and their interaction. And you can see from p-values all these factors as well as interaction, they are significant. That means both the factors are interacting with each other. And then we have another ANOVA table here, that means analyzing a particular factor within another factor. within the levels of another factor, and same for the other factor also. Then we have a final table, which is a very important table. That means this is the interaction table. Here you can see the average followed by the same lowercase letter in column and the uppercase in the row do not differ by 2 keys. Default p value less than 0.05. so we have a plot also here. We can see, like, we want to see the interaction plot, that is plot underscore interaction, then the name which we have given for this function. So we will click on this and we'll be having a plot here. This is the interaction plot, and these are the levels of the shades and these are the levels of the spacing, and you can find out that we have a significant interaction, whether we are having a significant interaction or not. So as far as the factorial experiment where factors are to be evaluated or to be studied in a particular experiment simultaneously in a single experiment with unequal equal precision, we have another type of factorial experiment that is known as split plot design. It is suitable for factorial experiments where large plots are required for application of different levels of one factor. So we have a data set for that. That is SPD, which is known as SPD here, is already imported. So we have a block variable here. This is actually the replications written here. Then we have a main plot factor and subplot factor, and we have a response variable. In case of split plot design, we used to have two factors. The main plot, it is to be tested with less precision in comparison to this subplot. And the important thing about this factorial experiment is that the randomization is done in 2 stages. We independently randomize this main plot, then independently randomize this second factor. So we have 2-stage randomization in case of the split plot design. But in case of a traditional factorial experiment which has got 2 factors with same or equal levels, the randomization is done in a single stage. So this is the big difference between these 2 factorial experiments. So in order to perform the analysis, we'll simply write names SPD. This is the name of my data set which is already available on my R console. So we have 2 factors, one is main plot and subplot. So in order to perform the analysis of split plot design within this R console, utilizing the library AgroR, we will write with a default function followed by the name of my data set, like we did in the Factorial CRD. Then we will use a default function which is PSUBDBC. This is the default function, and within that I have to write the name of my main plot followed by subplot. Then I have to write the name of the blocking variable. Here it is replication, followed by the name of my response variable, and in this data set the response variable is yield, and we can label out in order to display the graphical visualization of our results. Since the name of my data response variable is yield, so I will write it like in this way. This is a subjective approach. It depends upon what kind of target variable or what kind of study variable you are having in your data set. Then we'll click on this enter. Then again, like in the previous design, we were having the results. We'll be having the similar results in this case, and you can see here the Shapiro test in this example is found out to be significant. So the errors are not normally distributed. The distribution of error is not normal, and we have another problem here. As far as the Bartlett's test of homogeneity of variance is again found out to be significant, so the variants are not homogeneous, so there is no existence of homogeneity of error variances. And like we got the results of Factorial CRD, we have an additional information for coefficient of variations. We are having two, since we have two separate factors here. One is main plot and another one is subplot. Then we have an ANOVA table here, and you can see we have two different type of errors. One is for main plot and another one is for subplot. And again, there is an indication that your analysis is not valid, and it suggests try to transform your data. Since the error distribution is not normal, our first assumption is violated, so we have to do certain kind of adjustment, certain kind of transformation, which are very much required as far as the authenticity of the experimental design is concerned, because the heart of each and every experimental design is analysis of variance, as analysis of variance is based on certain assumptions which always evolve around the residuals or errors. So we have to be very confident, or we can say we have to ensure that those assumptions are fulfilled. Until and unless those assumptions are not fulfilled, we cannot proceed utilizing the traditional way of experimental designs or the parametric approach of statistics. So in this video we have demonstrated two different type of factorial experiments, one with equal precision and another one with unequal precision. Hope both these analysis will be quite helpful